Today, let's review that idea before we continue the further events during the human embryonic development. At the ampulo-isthmic junction, simultaneously when the male and the female gametes reach, there is fertilization or the syngamy. After which the zygote within the zona pellucida began the mitotic cell division, and that led to the formation of 2 cells, 4 cells, 8, 16, and that was followed by 32. And this multicellular body reached the uterus from the fallopian tube for implantation. Let's see a pictorial representation of what we have learnt already. This was actually the secondary oocyte, in fact, which was haploid, and that was collected into the fallopian tube, you can see there, and fertilization in the fallopian tube, and this one is a zygote actually, and that is all through the journey becoming the 2-celled, 4-celled, and the 8-celled, and finally becomes the 16, the 32, and this multicellular body which is enlarged here, reaching the uterus for implantation. What is the name of the cell which is produced after the mitosis called? The cell is called by the name blastomere. Let us see the multicellular body which is ready for implantation. The multicellular body which is ready for implantation will be having what kind of cells? This is the covering which is called as the zona pellucida. This I was emphasizing on the previous class also, and here you see a clear demarcation between the cells, the outer layer of cells. These outer layer of cells, which we call it as the trophoblast cells, and the inner group of cells which we already called it as the inner cell mass. This multicellular body is having outer trophoblast cells and inner cell mass. This will be visible in the uterus, and there will be formation of, let me fill up the cells completely, the inner cell mass. This will be definitely transformed into a next stage, which is the blastocyst. This blastocyst is identified by seeing a cavity, the presence of a cavity inside, and the zona pellucida, and of course the trophoblast cells, and the cavity like this. Group of cells here, the inner cell mass, and this is sometimes called the blastocyst cavity, the blastocele. At this stage, it is ready for implantation. Let's see how this process takes place. I am drawing the perimetrium, here the myometrium, and the highly developed endometrium. So when you draw this endometrium like this, highly developed because of the influence of the hormone called the progesterone being discharged from the corpus luteum. Now there will be an encounter between the zona pellucida and also the inner blastocyst. I am not drawing the cells here. This is the zona pellucida. You can see this will be definitely remote, and you can say this will touch with this and that will penetrate. What could be the entry point and which side will be into the inner and which one will be closer to the endometrium. Let's locate that point. See, after implantation, let's see how the structure is getting identified. This is the trophoblast, and here you will see the presence of that inner cell mass, and this one is that cavity, and the process called implantation is completed once this entire structure gets into the wall of uterus, that is the endometrium. Let us further examine the details about the embryonic development. The diagram presented before you here, you can see that after implantation, I have drawn the outer layer of cells called the trophoblast, and this one called the inner cell mass, and this multicellular structure with the cavity inside is implanted in the wall of endometrium. I have taken this body outside for explanation. Look at this. This one is the inner cell mass. The inner cell mass will experience one important process which is called delamination. D is removal, lamina a layer. So, a layer of cells will be removed from the inner cell mass. What is this layer and what is its future? After delamination from the inner cell mass, after delamination, we get the endodermal cells. These endodermal cells will form a huge bag-like body which is called as the yolk sac. Now, I have shown you here the structure called the yolk sac. So, after delamination, there is formation of an endodermal layer of cells which will proliferate, and that proliferation results in the formation of a bag which is called as yolk sac. Now, what do you call this multicellular unit which is remaining after the formation of the yolk sac? We call this as the embryonic disc. The embryonic disc, consisting of the embryonic cells, have to give rise to other germinal layers which include the mesoderm and also the ectoderm, proving that we are all triploblastic organisms. Let us see the pictorial representation. Multicellular body, and see this here, the inner cell mass labeled here, inner cell mass, and the cavity here which I was calling the blastocyst cavity, and the trophoblast cells, day 6, okay. And see, the next one clearly presents the yolk sac. Yolk sac, this is lined by this endodermal cells all over. That's clearly given as yolk sac there. And this one, the upper one, forming the embryonic disc. And this is on the day 10. And by then, the implantation is over. After talking about the formation of the yolk sac lined by the endoderm and the upper group of cells forming the embryonic disc. Since we are all triploblastic organisms, we will have 3 important layers, the primary germinal layers by name ecto, meso and the endoderms. Let me present before you the derivatives of the ectoderm, the mesoderm and the endoderm, so that a complete organism with all the organ systems and each organ having the required tissues will be structured within the human body. Here I have written ectoderm, ectodermal derivatives. The epidermis the outermost layer of the skin, which includes the stratified squamous keratinized epithelium, and nervous system is basically ectodermal in origin, and one cell in that, namely the microglia, will be derived from the mesoderm. The next one is called adrenal medulla. The inner region of the adrenal gland, called the adrenal medulla, is ectodermal in origin, and we all know it secretes a neurosecretion hormone by name adrenaline or the epinephrine. And let me remind you, during embryonic development there will be formation of nerve cord. Let's go into the second aspect. The second germinal layer is that mesoderm, one of the very important layers which transforms, catapults the structure to a higher grade of organization. Dermis, connective tissue. Connective tissue is a very hugely classified and well spread out tissue present in the nook and corner of the body. Musculature, the muscles. Of course, some muscles are ectodermal in origin. By and large, all the muscles are mesodermal in origin. The human endoskeleton, which includes the 2 0/6 bones, the adrenal cortex. The suprarenal gland's outer region is adrenal cortex, and that is regionalized into 3: the zona fasciculata, zona reticularis, etc. That one is from the mesoderm, and the lining of the body cavity. We all know eucoelomates, the body cavity is lined by the parietal and the visceral layers, and that is from the mesoderm. And endodermal derivatives, the lining of the gut, the columnar cells of the stomach, etc., are all endodermal in origin, and the pancreas, liver, the first, the second, these two are quite important during the process of digestion. Liver and pancreas, the largest gland of the body, and this will be the second largest gland of the body, and coming to 2 endocrine glands by name thyroid and the parathyroid. So these are the few examples to make you understand that the 3 primary germinal layers of a human body will give too many varieties of tissues, and they combine to form the organs, and organs combine to form the organ systems, and all the organ systems will make a human body. After learning the organogenesis and formation of organ systems in a human body from the various layers, ecto, meso and endoderms, let us understand the formation and the names of extra embryonic layers. Human being is basically an amniot. Amniots will have the development of amnion around the embryo, and not only amnion, we have the other layers too by name: the chorion, the amnion, the yolk sac, and the allantois. These 4 are termed as extra embryonic layers. Once we observe the formation of the derivatives within the embryo, we should also see the development of extra embryonic layers. The very name is suggesting outside embryonic, embryo layers which are present outside the embryo, named as chorion, amnion, yolk sac and allantois. Let's come into one of the very vital discussions, that is placenta. This important discussion should be learnt, like functions of placenta and the hormones discharged by the placenta and what makes the placenta, and based on what features we give the terminology required for the placenta. If you observe here, there is placenta, there is all a yolk sac, there is embryo there, and all these within the uterus. That means uterus, once the blastocyst is implanted, will shelter the entire structure within the endometrium all through the gestation period, that is the intrauterine development period. Here I am going to explain you the different parts of the implanted growing, well grown embryo, the fetus. Villi like finger shaped enormously growing structure, this one, these are called the chorionic villi. The chorionic villi are growing enormously deeply buried in the wall of this endometrium. Now, where from this chorion is growing? The chorion should grow from the outer most layer, usually the trophoblast. And not only the chorionic villi, even the uterine tissues do contribute. See that they get interlocked here, sometimes called interdigitated here. So this tissue is also going down, and this one is also clearly embedded there, and this entire structure now can be called as the placenta. This is called the placenta. And you will not see this villi all over, or rather enormously present everywhere. It will be very clearly present only at one point. Initially a metadiscoidal, but later the villi are restricted to one area and we term it as the discoidal placenta. Let us now study the functions of the placenta. Placenta has several functions, and few of them will be providing oxygen required to the growing embryo and also the nutrients. It will take away the carbon dioxide and the metabolic products. So we can say that the placenta will be a region where there will be exchange of material between the maternal circulation and the fetal circulation. The anatomy of the placenta. Here, look at this word, maternal arteries. See how they are moving in like this, okay? And the veins going away, the maternal veins. And what about this one? Highly branched, the red and the blue indicating the arterial and the venous. And here also, what is this entire structure here? This is the placenta. And please note that the maternal blood will not mix with fetal blood. If this happens, it will prove detrimental for the growth of the young one, because the maternal blood not only supports the young one, it proves detrimental because it contains female hormones and it will have metabolic byproducts and other molecules related to the mother. So there is no question of the maternal blood entering or mixing into the fetal blood. This is one of the notable points. It is a 𝗧𝗦𝗔𝗧 placenta. So already we have used one word before, that is discoidal, and now we can use the word 𝗧𝗦𝗔𝗧 placenta. And there is no, I repeat myself, there is no mixing of the blood between the mother and the next generation in making. Let us study now the various hormones that are secreted by the human placenta. Human chorionic gonadotropin, called the 𝗧𝗦𝗔𝗧. This is released by a group of specialized cells which are called the 𝗧𝗦𝗺𝗺𝗺𝗺. Understand the word that 𝗧𝗦𝗺𝗺𝗺𝗺, that means the outermost layer which we were speaking before, the trophoblast layer, it will get modified into syncytiotrophoblasts, and that syncytiotrophoblasts are the group of cells which will release this hormone called a human chorionic gonadotropin. Human pregnancy confirmation is done by this particular molecule called hormone called human chorionic gonadotropin. And let's see the second one, human placental lactogen, abbreviated as HPL. It is also called somatomamotropin. This particular hormone, the HPL, will have anti-insulin effects. So when there is anti-insulin effect, automatically there will be increase in the sugar level or sustained glucose level, and that makes the glucose available to the fetus. And that's very vital for the fetus because of this hormone called the human placental lactogen, somatomamotropin, having anti-insulin effects. There will be hyperglycemic state in the blood plasma, providing the glucose for the developing young one. A third one called estrogens and the progesterone. This makes us understand that placenta is a temporary endocrine structure which produces too many varieties of hormones, and I have listed about 4. Human chorionic gonadotropin, the human placental lactogen, the estrogen and progesterone, these 4 released from the placenta. Let us now understand what are those peculiar hormones released in a human female during the course of pregnancy. These 2, human chorionic gonadotropin and human placental lactogen, together with relaxin, these 3 will be only during the gestation period. And during the menstrual cycle, we do find estrogen and the progesterone, and we have discussed this before in our topic menstrual cycle, wherein the estrogen was more during the first 14 days, and after which there was increase in the progesterone level. Let's see the various events during the gestation period. We have learned the various events right from the fertilization, implantation, organogenesis, and placenta functions, secretions of the hormones from the placenta. Let's divide the intrauterine development, the gestation period, into 3 trimesters, each one include 3 months, trimester, 3 months. Let's see what happens during this first month, second month, and the third month. We will see the development of, during this, we will see the development of, you can see very clearly the brain, formation of brain. It means the ectoderm has given rise to the brain, the nervous tissue, very well developed within this, and heart, blood vessels, major internal organs will be developed during the first 3 months. And what about the umbilical cord? You can see, in addition to the placenta, we also see an additional structure which is I'm pointing out there, that's called umbilical cord. So let's review the events which are happening in the first trimester. Heart formation, blood vessels, indicating there is circulation in the circulation and heartbeat also does begin. Major organs are formed, including the genital organs, the reproductive organs. After the first trimester, where we have seen the formation of the major structures of the body, the genital organs, and also especially the umbilical cord, the link between the young one and the placenta, from which you can see the umbilical cord. Now, what about the limbs? The limbs, because we are all tetrapods, we need to get the forelimbs and the hind limbs. As this picture is clearly depicting, you can see the growth of the forelimbs, the hind limbs, and here also you can see the forelimbs, and you can see umbilical cord also slightly here, and also the hind limbs too, forelimbs, hind limbs, the umbilical cord. What about the next trimester, 4, 5 and the 6th months? We can see this is the 4 and the 5 and the 6th one. You can see an enormous growth. What is very important is when the young one is growing in the uterus, naturally the dimensions of the uterus must also increase to accommodate the growing fetus. In the second trimester, there will be movements of the fetus, and there will be formation of the eyelids, the eyelashes, and of course the growth is certainly progressive. And say this, the limbs are also increasing in their length. The umbilical cord is very clearly visible now, umbilical cord here, again the forelimbs, the hind limbs, and the movements of the fetus. We have seen the second trimester, the events during the second trimester. So if you start from the first and the second, and by the end of the 6th month gestation period, we have seen a huge transformation. We have seen the single cell structure growing enormously, it has become multicellular, or tissue growth, the organogenesis, and the organism growing through first, second, third, ultimately up to the 9th. So when we started with the first trimester, we have seen some major changes, heart, blood vessels, and the second one, we have seen the eyelids, the eyelashes, and we all know mammalians are identified by the presence of the hair. The fine hair could be seen, the growth of the fine hair could be seen during the second trimester. And what about the final trimester, which includes the last 3 months, the 7, 8 and the 9. You can see how the organism is slowly, gradually, a small change in the positioning. Probably the head is reaching this cervix region of the female. Probably the young one will respond to the noises, the sounds, etc. And movement of the fetus is already finished in the second trimester. So by the time we come to the final trimester, we see complete growth of the young one ready for parturition. Let's see the final event, which is called the parturition. All through the journey of the gestation period, we have seen the growth being very magnanimous, very complex, and that's the journey of life. And when the next generation needs to come out from the mother's womb, the uterus, there is a complex mechanism which we are going to learn now. How does this event called childbirth, parturition happen? Before we go into that event called the parturition, look at this. Completely grown young one at the end of the 9-month gestation period, the head is closer to the birth canal. As you know, the cervix and the vagina, they're together called the birth canal. We need to see changes especially here so that the young one is expelled out of the uterus. How does this happen? It all starts with movement of the fetus. A fully grown fetus together with the placenta will initiate this total event. A fully grown fetus and the placenta, there is a movement in the fetus resulting in fetal ejection reflex. Fetus, placenta together cause this particular change, and the fetal ejection reflex leads to what? This fetal ejection reflex, the signals are reaching the pituitary gland, and the neurohypophysis of the pituitary gland releases the hormone called the oxytocin. The oxytocin reaches the myometrium of the uterus and initiates the contraction of smooth muscles in the myometrium of the uterus. The strong contractions further trigger the production of oxytocin. So it's like oxytocin acting on the smooth muscles of the myometrium of the uterine wall, and the smooth muscles contracting, and contraction further initiates the release of the oxytocin. So there's a contraction there, release here. So as we go step by step in this process, the contraction becomes more and more powerful, more and more vigorous. More the contraction of the smooth muscle, more is the discharge of the oxytocin. Increase there, increase here, muscle and the hormonal discharge. Ultimately, the oxytocin levels increase, powerful muscular contraction of the uterine wall, and that leads to the expulsion of the fetus, the young one, through the birth canal, that is cervical canal, and the vagina. Ultimately, the young one is delivered, and this is what is called parturition. What about the placenta? During the parturition, the placenta should be removed. That's the reason why we call it as deciduous placenta. We have seen the birth of a young one, which is termed parturition, which was a neuroendocrine mechanism, a complex one, which was involving the fully grown fetus and pituitary gland, which is releasing the oxytocin. We have observed the oxytocin being released more and more, and the movement is more and more, and because of this, there is parturition. What even follows next is lactation, the milk discharge or release from the mother. How is this possible? We all know that the human mammary gland will be the one which is discharging the milk. We have learned the sequence too in the previous classes. The alveoli, the mammary ductules, the mammary ducts, the lactiferous, and when we have learned that a mammary tissue is meant for lactation, what are the hormones which are making the mammary glands to deliver its function? I have written here the 4 hormones: estrogen, progesterone, prolactin and oxytocin. The very first one, estrogen, growth of mammary tissue. That is vital for developing the cells which can help in milk production. So, the growth of mammary tissue is done by the estrogen. What about the second one? Progesterone. Progesterone will be causing the development of receptors, prolactin receptors on the cells of alveoli present in the mammary gland. We call this priming. And we all know estrogen and progesterone are not only released by the ovary during the course of the menstrual cycle, they are also released by the placenta. So that is a very clear indication that estrogen and progesterone can come from two different sources, one being the ovary and the other being the placenta. What about prolactin? Prolacto, the one which supports the production of milk. So prolactin causes the milk production. And finally, ejection of milk is done by the oxytocin. So 4 different hormones are required for this process of making the milk, ejection of the milk. We call this sometimes a synergistic effect. After understanding the effect of various hormones like estrogen, progesterone, oxytocin, prolactin on the mammary glands, let us study now the composition of milk. The releasing of the milk is called as the lactation, and the ejection is done by oxytocin. And in the initial days of milk discharge, it is termed colostrum. So colostrum is the milk released by the female in the initial days of lactation. What are the various components of colostrum? Colostrum is heterogeneous. It has too many components. Some of them let us examine. The lactose, lysozyme complements lactoferrin, immunoglobulin A, G and M are also possible, and proline-rich phosphates, etc. When we study the composition of the colostrum, it's proving that there is presence of antibodies in that. So, breastfeeding in the initial stages of human development, we have learnt how 4 different hormones are responsible for the growth of the mammary gland, mammary tissue, the priming, the release of the milk, and finally the ejection of the milk. Mother during the initial stages will release the milk. The releasing of the milk is called as lactation. In the initial days, the release of the milk, and the milk is called by the term colostrum. What are the various components of colostrum? It is heterogeneous in nature. It has got lysozyme, lactoferrin, immunoglobulins, various types of antibodies, and then this proline-rich phosphates, etc. Let us learn now how the presence of the antibodies help the newly born. This is a part of passive immunity, natural passive immunity. We have studied how 4 different hormones are acting upon the mammary glands in a human female, transforming that structure into highly functional structure, functional body. Let us now understand the terms lactation, colostrum, its composition, and the required terminology. When we say the word lactation, the milk production in a female can be called as the lactation. When does the female begin to lactate? By the time she enters into her 3rd trimester, the ending of the gestation period, there will be a preparatory activity for lactation, and lactation does commence. And after parturition, it is absolutely necessary to feed the infant. The milk that is released during the initial stages of lactation, I repeat, the milk that is released during the initial stage of lactation is called as the colostrum. Let us now study the composition of the colostrum, the milk released during the initial stages. Lysozymes. We all know this is a very important bacterial enzyme, and the complements. I remind you, this is a soluble mediator of immunity, the complements, and the lactoferrin, which protects the lining of the gut from bacteria, immunoglobulins, the antibodies. So certainly, consumption of the milk by the infant is very important to prevent infections and to protect the newly born infant. The heterogeneous in nature, the colostrum is heterogeneous in nature because it has too many components in that. So, newly born infant should be breastfed to prevent infections and to improve the immunity of the young one. Dear students, we have seen the entire course of event, which includes fertilization, embryonic development, 4, 6, 8 cell stages, the implantation, the formation of placenta, umbilical cord, and the various extra embryonic layers, chorion, amnion, yolk sac, and placenta, the 3 trimesters, and after which there is parturition, and the young one feeds upon the mother's milk, which is called the colostrum. All through the gestation period, the female is metabolically very active because the placenta is releasing specific hormones during the gestation period. In addition to those which are coming or released from the placenta, the human female body during the course of pregnancy will have release of several other hormones. Let's examine them. I have written 3 here: cortisol, prolactin, and the thyroxine. Cortisol, a very essential hormone, steroidal hormone released from the adrenal cortex of the mother, and prolactin from the pituitary gland, and thyroxine. We are all familiar with the thyroxine iodinated hormone, which can improve the basal metabolic rate. That means a human female during the gestation period, when she is a pregnant woman, will have release of a very diversified form of diversified hormones in her body. I am just giving you a model here. When we revise the human development, we need to focus on some of the events which happen in a chronological order, and that will be one of the possible questions, sequence questions. I just took a model here. Arrange the following in a correct sequence: blastocyst with zona pellucida, morula, yolk sac, and the zygote. When we see the sequence, the first one will be the zygote. Zygote, the diploid cell formed at the ampuloisthmic junction, that will be the first one. And then you all know it becomes the 2-cell stage, and the cells are called the blastomeres, and that becomes the 4, 8, 16, 32, and that moves into the uterus. So the morula will be the next one, and that is followed by the blastocyst. I wrote one word called zona pellucida. We know that blastocyst will have zona pellucida in the beginning, and later on it will lose the zona during the course of implantation, after which it will develop this yolk sac. So the sequence will be 4 will be the first one, then we can see the second, and then the 1, and finally the yolk sac. That means third will be a right answer. Let's examine one more why it could not be. Let's see the first one. 1, the blastocyst, and then the second one is morula, which is definitely incorrect. Morula will be followed by the development of or the formation of the blastocyst. What about 4 and 3? 4 is zygote, and immediately you will not see the formation of the yolk sac, so that is also eliminated. What about the final one? 2 and 1. 2, the morula, and then the blastocyst. This seems okay, but what about the 3? The yolk sac. But 4, zygote at the ending? Absolutely incorrect. So the only possible answer is 3. Let's see another question. Hormones released only during the gestation period are The first one, human placental lactogen, growth hormone and prolactin. This option will be incorrect because growth hormone is produced all through the life. And the second one, HPL, 𝗳𝗏, human chorionic gonadotropin, progesterone and insulin. In this set, insulin is the one which is the wrong pick because insulin is required whether the female is pregnant or not. Insulin is must to maintain the carbohydrate metabolism. What about the third one? HPL, okay, but insulin and thyroxine will be incorrect because these two are must to maintain the sugar level and also the metabolic rate in a non-pregnant female. And what about the last one? When the three options are incorrect, fourth will have 100% possibility to be the right. Let's see. HPL, 𝗳𝗏 and relaxin, and these three are only during the gestation period. So this will be the right answer, fourth one. Let's see another question. I wrote it as, which one of the following is not released from placenta? 𝗳𝗏, HPL are released from the placenta. HPL, estrogen released. Estrogen and progesterone released, and these two are not from the placenta. Glucagon is from the alpha cells of pancreas and the cortisol is from the adrenal cortex. So fourth will be the right answer when we understand about the hormones which are released from the placenta will be 4. Not released is the point. So not released. So these two are not released from the placenta. The statement is colostrum released during initial stages of pregnancy is required to protect infant. Statement 2, colostrum contains immunoglobulins. See, this statement can be at times confusing, or rather at that nick of moment probably we can go for a wrong answer. The statement clearly tells colostrum released during initial stages of pregnancy is a wrong statement because a pregnant woman begins to release milk or she begins to lactate at the ending or at the final stages of gestation period. And here the statement says colostrum released during initial stages of pregnancy. This statement 1 is false, is incorrect. Statement 2, colostrum contains immunoglobulins. That is the right statement because immunoglobulins are antibodies. We can see the presence of one of the antibodies in the colostrum. So statement 1 is incorrect because of the line initial stages of pregnancy has nothing to do with colostrum. So statement 1 is incorrect and statement 2 is correct. Please do focus on such lines to evaluate statement type of questions. The last question based on the diagram. I have given 4 different diagrams here. Each one is indicative of one particular event. Look at the first one, A. The sperm is reaching the secondary oocyte and there is a sign of fertilization there. And the second diagram, 2 cells are present. The zygote divides by mitosis and we get 2 cells. After the first cleavage, the zygote will give 2 cells and we call it blastomeres. And the third one, now it is having that zona pellucida, that means not yet implanted, and is having the outer trophoblast and inner cell mass here, the inner cell mass containing definitely some stem cells also, and that is a multicellular body, maybe around 8 to 16 cell stage. If this has to be arranged in a sequence, let's say the first option, A, okay, a fertilization process where the sperm is entering, that is secondary oocyte, and B is this 2 cell stage. C cannot be a sequence because it is having this outer trophoblast and inner cell mass, and that comes only after the multicellular stage. The second option is A. Yeah, A is syngamy there, fertilization, and B, the 2 cell stage, the blastomeres, and D is that 16 to 32 cell stage, and the C with a clear differentiation between the outer trophoblast and inner cell mass. The 2 will be the right answer. A and the B, 2-celled, the D, the multicellular, and the C, which is with outer trophoblast and inner cell mass. So dear students, please do revise this topic, especially for the order, the sequence questions, and terms like colostrum, and do not go wrong with the word cholesterol, and the hormones released from the placenta, and which hormones are more during the pregnancy, which are generally found even in the normal non-pregnant woman. So demarcate between the placental hormones and the hormones which are released from other endocrine glands of a female during pregnancy, and match the following questions, diagram based questions and statement. Read the complete line and see whether the word is correct, and check for the word not, and some of the word not is given somewhere. Please try to read the preceding and succeeding lines before and after the word not. And that brings to the end of the topic called implantation, embryonic development, the placenta, the lactation, and please do focus on important terms.